classification system. And right now, like we have done 2 classes already. Okay, so we have done 2 classes already, and in these 2 classes, we did Monera and some part of the Protesta. So before starting the session from the 7:30, I want to recap whatever we did. Fine. So Yasir, okay. Now you are able to unmute yourself. So tell me, quick, what you did in your school? Which chapter? You can text me as well. No issue, because I'm unable to listen you. Fine. So and today you will come to know how we are teaching, like what is my methodology of teaching. Look, we have started from the biological. In case if you are feeling that you are facing any problem anywhere, like in terms of voice, in terms of pace, in terms of any clarity, so you can let me know, right? Feel free to ask me anything. Fine. So actually we have started with the classification system, this biological classification system, right? Cell unit of life, cell cycle and cell division are done. Okay, biomolecule is going on. Oh, they have started from the cell. Fine. So, before starting any chapter in this syllabus, first we must be aware with the classification system. Unless you are not aware with the classification system, the syllabus have been designed in this way that the first chapter will always come first, right? So this is a sequence of learning, right? So we have to start it from the biological classification part, and we have, in fact, we have started with that. Fine. So try to understand. If you want to note it down, you can, but I will suggest you don't note it down right now. You will get the link of these PPT, whatever I have written on the whiteboard, and after the class, you can go with that. Right now, I'll try to explain you. Look. So the first and foremost classification that was given by a scientist, Aristotle. So he is a father of biology. So the father of biology, Aristotle, he said that there are 2 types of the animal in this world, right? Why I am telling you, because this question have been asked in many examinations, and uh, if you will in future, if you will prepare for the competition, these kind of the things which are missing in the NCERT. So I am trying to enrich with this your NCERT. Fine. Look, he is called father of biology. So according to Aristotle, but Aristotle given this concept 350 BC, means from today or approximately 2500 years ago, right? Means 350 years before the birth of the Christ. So there are 2 types of the animal in this world: those which have red blood and those which do not have red blood, right? So with red blood and without red blood. He said that there are 2 classes of animal, and those who have the red blood, he called them in aima, right? And those which do not have red blood, he called them anaema. So Aristotle classified animal in 2 categories: enaema and anaema. So which are the animals which do not have, who are those animals which do not have red blood? For sake of octopus, so need to remember that octopus always have blood, blue blood, right? Insect, they do not have blood, right? Colorless blood. Crab, they do not have blood, right? That is a colorless blood. Fine. Then the next classification system was given by Carl von Linnaeus, right? He given 2 kingdom classification system. Now the classification from the NCERT start from here only, right? So he divided animal living into 2 parts: kingdom Plantae and kingdom Animalia. In Plantae, he kept the plant. In Animalia, he kept the animal, right? What was the criteria behind classification? How did he separate it? How did he separated plant from the animal? In the present, first criteria was presence of cell wall. That is important. This can be question for your school exam. the criteria behind dividing two kingdom classification system by lineage. That presence of cell wall, plant have, animal do not have. Presence of inorganic crystal, plant have, animals do not have. Presence of vacuoles, they are found in the plant, not found in animal. Nutrition, plant can make their own food, but animal cannot. Presence of organs, plant do not have any organ, like excretory organ, right? But animals do have the organ, right? So these were the 5 criteria behind dividing the two kingdoms. After some time, in 1866, there was a scientist, his name was Ernest Haeckel, right? Ernest Haeckel said there are third kingdom. So existence of the third kingdom. He said there are 3 kingdom classification systems. So he added on the third kingdom, and that was the Protista. So he said, along with the Plantae and Animalia, he added the third kingdom. What he did, he separated all unicellular organism from multicellular organism. He taken out all unicellular animal, unicellular plant, and unicellular fungi, right? And create a third kingdom, Protista kingdom. That's why it is also called a mixed kingdom. Then it comes the 4 kingdom classification. Before 1956, the discovery of electron microscope had not been done. So at the advent of electron microscope, when electron microscope discovered, on that time they came to know there are 2 kinds of the cell, prokaryotic cell and eukaryotic. So Copeland said that prokaryotic cells should be separated from eukaryotic cell, right? And he created a new kingdom for all these prokaryotic organisms, right? So kingdom Plantae was already there, Animalia was already there, Protista was already there. He created, means Copeland created a 4th kingdom where he kept all unicellular prokaryotic cells, right? And created a 4th kingdom Monera, right? So he kept all the prokaryotic organism in Monera like bacteria. So this was the classification system from 2 kingdom, 3 kingdom, 4 kingdom, right? And then we will start with the 5 kingdom classification. Is that clear to all of you? I'm just recapping you. Is that clear to all of you? Are you getting it? Yasir?
Yes, sir.
Nabiya? Okay, great. Fine. So look, now come to the 5th kingdom classification system, right? I'm just right now I'm in a pace, right? I'm just speeding it up because I want you to get the recap or get the conclusion of before I start the class. Then you will enjoy the class. In the class, my pace will be a bit slow, right? So just right now, just you have to focus and try to understand whatever I am delivering, right? Then it comes 5 kingdom classification. So nowadays we are studying the 5 kingdom classification. This 5 kingdom classification system was given by R.H. Whitaker in 1969. So there was a scientist, his name was R.H. Whitaker. He given a 5 kingdom classification system. So what was the criteria? How he designed these 5 kingdom classifications? All these 4 kingdom were existing already. He just redesigned this system and added on a 5th kingdom, fungi, right? Before this 5 kingdom classification system, fungi used to be studied in the plant classification, right? Plant part means plant kingdom. According to Whitaker, he said there are 2 kinds of the organism. Prokaryotes, mean which are made up of prokaryotic cell, and eukaryotes, which are made up of eukaryotic. All the prokaryotes are unicellular; they can never be multicellular, right? So he kept all the prokaryotes in Monera, and that is represented by bacteria. Then he said eukaryotic cell can be of 2 types. They may be unicellular and multicellular. So all the unicellular eukaryotic were kept in the 2nd kingdom. That is the Protista. Then he said multicellular organism can be of 2 types, which have cell wall, which do not have cell wall. Plant and fungi have cell wall, and animal do not. So he created a 3rd kingdom, Animalia. Now with the cell wall. So with the cell wall, there are 2 organisms, fungi and Plantae. But fungi are not green; they cannot make their own food. They are dependent on dead and decaying material. So which do not perform photosynthesis, fungi, and which are able to perform photosynthesis. That's how he created 5 kingdoms. 1, 2, 3, 4, and 5. Right. So this was the 5 kingdom classification system designed by the R.H. Whitaker. You can get the question from this part as a long answer, right? Long question in your school examination. So that can be the question. Describe the 5 kingdom classification system and what was the criteria behind designing the 5 kingdom classification. So what was the criteria behind devising this 5 kingdom classification system? First, first criteria was complexity of cell. Prokaryote and eukaryote. Prokaryote are simple and eukaryote are complex. Then complexity of body structure. Unicellular, multicellular. Third, mode of nutrition. Photosynthetic like plant. Absorptive nutrition like fungi. Ingestive nutrition like animal, right? Autotrophic like plant. Fourth, ecological lifestyle. There are some consumers like animal, like protista, or producer like plant, or decomposer like fungi, right? So that's how that was the ecological lifestyle. Fifth criteria was phylogenetic relationship. Phylogenetic relationship means evolutionary relationship. Who came first? Who came later? Those organisms who came first in the line of evolution, they kept first, like Monera and Protista. Those who arrived on the earth in the last, like Animalia, they have been kept in the last. So these are the 5 criteria behind designing the 5 kingdom classification system. Now, 6 kingdom classification system was proposed. It do not exist. We do not follow 6 kingdom classification system. Just 5 kingdom classification was given, and we follow the 5 kingdom classification today, right? But so, what is the 6 kingdom classification system? Just a minute. So he designed a 6 kingdom classification system. What is the 6 kingdom classification system? That was proposed classification system, right? So what was the proposed classification system? It was proposed in 1990. Carl Woos was a scientist who said that Monera should be divided in 2 parts: eubacteria and archaebacteria, right? So Monera is represented by bacteria. Look, this Monera is represented by bacteria, right? Carl Woos said there are 2 kinds of the bacteria: eubacteria and archaebacteria. Archaebacteria are the oldest cell. They are the oldest bacteria. And eubacteria are new bacteria. Eu means new and archae means old, right? He said that archaebacteria should be separated from Monera and that should be kept in the 6th kingdom, right? So, but this was the proposed. It have not been accepted. Now tell me, is that clear or not? If you have any question, please ask me. 5 kingdom classification. Yasir, is that clear? Nabia? Great, great. Nabia? Haya? Is that clear?
Yes, sir. Yes, sir.
Very good, very good. If it's clear, it's a very good to listen, because it means you are getting my points. So now I have started with I have taught them, look what I have taught them. I have taught them Monera and I have started with the Protista, right? So look, Monera kingdom is represented by bacteria, right? The experimental organism in bacteria, what we study, that is the E. coli, Escherichia coli bacteria, right? So this is the experimental organism which we study in our classroom program, right? So this is the bacteria, and just I'm going to give you recap. I'll teach you again. I'll give you some extra class someday, right? Today it's just recap, right? I'll give you some extra class to cover it, right? I'll again explain it in detail. Today just I'm going to recap you, right? So don't think that this is the proper class, right? So look, here we have kept the bacteria. Bacteria is a prokaryotic organism, right? It's a DNA means it do not have nucleus. It is always unicellular, right? Their ribosome is smaller. They multiply by binary fission, right? So here we have kept the bacteria in Monera, right? So I have taught the bacteria. So then archaebacteria are older bacteria, which are older than this bacteria, and they are found in very hostile condition where other cell cannot exist, right? They are found in the mouth of the volcano, right? They are found in the deep oceans. They are found in the sulfur rocks. They are found in the uh, salt lake. They are found in the methane gas, presence of methane gas. So where the other organism cannot survive, they are found. So they are found in extreme and momental conditions, right? And they are the oldest cell of the world. So in Monera, we study the bacteria. We studied the archaebacteria. Then we study the blue-green algae. Blue-green algae are also called cyanobacteria, right? So what they do, actually, their specialty is that they fix atmospheric nitrogen into soil. Means they capture the nitrogen of air and they convert it to nitrogen of the soil nitrogen, right? And the last organism is mycoplasma. This is the smallest organism of the world. It do not have cell wall, right? So in Monera, what we have studied in kingdom Monera, bacteria, archaebacteria, blue-green algae, and mycoplasma. I will explain all these four in detail in some extra class, right? Then I started with this kingdom, what I am teaching right now, right? Then I have started with the kingdom Protista. So in kingdom Monera, we have just kept what the prokaryotic organism, right? In kingdom Protista, we have taken the unicellular eukaryotic organism, right? So unicellular eukaryotic organism. So kingdom Protista is called mixed kingdom. That can be a question for your school examination. Why kingdom Protista is called mixed kingdom? Try to understand. Look, we have taken out all unicellular eukaryotic cell from plantae, from fungi, from animalia, right? So from plantae, we have taken out the unicellular algae. From fungi, we have taken out the unicellular fungi. From animalia, we have taken out the unicellular animals, right? So we have taken out unicellular organism from all kingdom and we have kept under a single kingdom Protista. That's why it is called mixed, right? So these are some general characteristic of Protista. Then type of Protista. How many type of the Protistans are found? So on the basis of nutrition, we have divided Protista into three kingdoms, right? Into three parts, right? First, photosynthetic Protistans, those Protistans which can make their own. Then consumer decomposer, those Protistans either they eat something or they decompose something, right? Here comes to the fungi slime mold. Then protozoan Protistans, those which are from the animal background, right? Those which are the animals, right? So that's how we have designed the type of Protistan. In the type of Protestant, we have started this studying in the last class in our regular batch. We have started with the photosynthetic Protestant. That is the algae. So these have taken from the plant, right? In photosynthetic Protestants, so further we have divided photosynthetic Protestant. It is all given in your NCERT. So in photosynthetic Protestant, we have divided them into three categories again: dinoflagellate, chrysophyte, and euglenoid, right? So photosynthetic Protestant, they are called phytoplankton. What are the phytoplankton? Can anybody tell me what are the phytoplankton? Yashir, Nabiha, Haya. What are the phytoplankton? These are the small cells and these fishes eat them. Fishes and small animals, crustaceans, they eat them, right? And they are green photosynthetic organisms, right? So general characteristic. So look, first I have started with the dinoflagellate. So this is the structure of dinoflagellate. It is given in your NCERT book, right? So this is the structure of dinoflagellate. Dinoflagellate have two flagella. You can see this vertical flagella and this is horizontal flagella. This one transverse flagella. They have two flagella in 90 degree, right? One horizontal, one vertical. Then this is apex. They have reserve food, starch, oil, and this girdle-like structure. This girdle, horizontal girdle, right? It is known as the cingulum, right? And it have larger nucleus. So they are mostly, what are their characteristics? They are mostly marine, but some are found in fresh water. Mostly they are found in the ocean. This name you have to remember. Gymnodium is a type of dinoflagellate, and Goniolax. You can see this is the Goniolax and this is the Gymnodium. These two names you have to remember. They are most frequently asked questions. Gymnodium and Goniolax, they make water red and called red tide phenomenon, right? So, like you see the Red Sea. What is why it appears red? Red Sea appears red water because they have these red kind of the microorganisms, right? So that's why the water appears red. Some of them show bioluminescence phenomenon. Means some of them they produce light, right? In the night time they emit the light. That's why water appears sparkling at the night time, right? So that is called bioluminescence. Bioluminescence when some biological organism emit the light. Aisha, I am just giving the recap to these new students, right? And you was there in last class? I think you was there in last class. I'm unable to listen you. Tell me, Aisha. Was you there in last class? Hello. Oh, you was not there in last class. So it's also for you. Last to last class, was you there? Or it's or this is the first class for you? Okay. So you are late, Aisha. We have started from the 7 o'clock, right? 7:05. Aisha, we are studying the classification system, right? Five kingdom classification system. In five kingdom classification system, right? We have started with the this one. We did this Monera, right? Then now we are doing the Protista. In Protista, there are three kinds of the Protista: photosynthetic Protista, consumer decomposer, and protogen Protista. So we are doing the photosynthetic Protista, right? So they are also called golden brown Protista because their color is golden and brown. They have two flagella, I have told you. They contain the chlorophyll A and C, means chlorophyll B is not found, which is found in the plant. The starch and oil is reserve food. Asexual reproduction by cell division. Sexual reproduction in present some forms, not in all. Right? Then look. So first, photosynthetic protistans are dinoflagellate. Second, chrysophytes. So today I have to start from the chrysophyte. Tell me, photosynthetic protestant, is it clear or not? Aisha, are you getting me? Okay, fine. Aisha, are you getting me what I am teaching right now? Actually, four of you, like Yashir, Nabiha, Haya, and Aisha. This is the first class for you, right? So I was just today. I have started this class at the 7 p.m. By the way, the class timing is 7:30 onward. But today I have started this class from the 7 o'clock to give you recap what I am teaching. But I saw you joined us late, right? But in the meantime, I want to tell you those who have joined us today, right? I will give them backup class to cover all these things in detail, which I have already taught to our older student like Martin and all that, right? In the meantime, tell me, Yashir, Nabiha, and Haya, did you got this thing? Did you got an idea what we are studying or not?
Yes, yes.
Okay, I'll give you. Don't worry, I'll give you backup class in detail. Right, Martin. So from where we have to start today? Tell me. Aisha, did you got some idea that what we are doing? Okay, great. Martin, from where we have to start today? You cannot speak. Okay, I have given per rights to all of you to speak. You can unmute yourself, but let me recheck it again. Yes, Martin, try to unmute yourself. Is it working? If not, then you should rejoin because other are able to speak. Okay, so Martin, are you able to speak now? Okay. Your microphone is not connected. Connect your microphone first. Martin, are you able to listen me? You can message me as well. Yes, Martin, unmute yourself and let me know. Are you able to unmute yourself right now? No. Let me ask my team. But what about the rest of the one? Aya? Aisha, are you able to unmute yourself?
Yes, sir.
Okay, Martin, I think some problem is at your end, but even let me. In the meantime, Martin, you can message me what we did in last class. As per my record, we did this one. We did this one. Dinoflagellate. So we have started with the. Let me recap. We were studying the types of the food protistans, right? So in the last class, we discussed that there are three types of the protistans. Photosynthetic protistans, those protista which can. So what are the protistans? Unicellular eukaryotic organism, right? Unicellular eukaryotic organism, right? Before starting that, I want to know, are you aware with the unicellular and eukaryotic and prokaryotic cell? All of you, are you aware with that or not? Yashir, aware with that? What is the difference between unicellular, eukaryotic and prokaryotic cell? Yashir, Nabiha, Haya, Aisha, even Martin. Okay. Guys, first we need to know what is the difference between Unicellular, what is the difference between eukaryotic cell and prokaryotic cell? Look, so before starting the biological classification, let me add on these.
Sir, good evening, sir.
Very good evening, Shadika. What happened? You are late today?
Yes, sir. Sorry, I had to open my laptop and set it up.
Okay, okay. Eukaryotic. So, Shadika, are you aware with the difference between eukaryotic and prokaryotic cell?
Yes, sir.
So, rest of the people are not clear with that, right? So, let me first tell them what is the difference between eukaryotic and prokaryotic cell.
Okay, sir.
Yes, Martin, that is true. Eukaryotic have. So, let me give the brief to all of you. Just a minute. What is the difference between eukaryotic and prokaryotic cell? Give me just 1 second. I just want to make it simple for you. In fact, I will. Look, try to understand, guys, what is the difference between eukaryotic and prokaryotic cell? Simply, the first cell which arrived on the earth, that was the prokaryotic cell. So what does it pro means? Pro means old, right? And there is a term carry-on, right? Carry-on or karyotic. Carry-on means nucleus, right? So those organisms which have older nucleus, they are called prokaryotic cell. Latin word eu. Eu means always, it means new, right? And again, there is a term carry-on, right? Carry-on term is nucleus. So those which contain, yes, Martin. Those which contain new nucleus, they are called eukaryotic cell. And those which contain old nucleus, they are called prokaryotic cell, right? Now try to understand. Look, in prokaryotic cell, this is a prokaryotic cell. It's a bacterial cell. Bacterial cells are always prokaryotic cell. Try to understand. In prokaryotic cell, look, this is a prokaryotic cell where DNA is present, right? But this DNA is not surrounded by any nuclear membrane, any cover, right? Okay, DNA is not surrounded by any nuclear membrane. That's why we do not use nucleus term for it. To call it nucleus is wrong notion, right? What we can say, we use a term that is called nuclei, right? I have already discussed with our regular student. So nucleoid, we use the term nuclei, right? This is just DNA. It is floating on the uh, protoplasm, right? So that is called prokaryotic cell. Here you can say there is a nucleus, right? So there is a nucleus. There is a structure which we call nucleus, right? This complete structure is called nucleus. To be called a organelle nucleus, there should be three criteria. There should be three eligibility. First, there is a small nucleolus. The gel which is filled inside there, that is a nucleoplasm. They contain a DNA, right? And again, there is a nuclear envelope, right? Here in prokaryotic cell, you can say there is a no nuclear envelope, there is a no nucleoplasm, there is a no nucleolus, just there is a DNA which is floating in protoplasm, right? And there is this nuclear envelope. It divide protoplasm from nucleoplasm, is separate nucleoplasm from protoplasm, right? So these kind of the cell which are evolved cell where the DNA is covered with nuclear membrane or nuclear envelope, and there is a nucleoplasm and protoplasm is separated. This kind of the nucleus is called new nucleus or eunucleus, and these kind of the organism are called eukaryote. So which are the eukaryote? The bacteria are prokaryotes. So this is the example of bacteria. So bacteria are prokaryote, right? And I have already told you that all monera, all organisms which come under monera, they are the prokaryote. Which are the eukaryote? All the plant, right? All the plant are eukaryotic, made up of eukaryotic cell, right? All the animal, they are made up of eukaryotic cell. All the fungi, they are made up of eukaryotic cell. All the protistans, right? Protista, they are made up of eukaryotic cell, right? But the only difference is there, plant, animal, and fungi, they are multicellular, right? Multicellular means they are made up of more than one cells. But protista, they are unicellular, right? But they all are eukaryotic cell. Here, bacteria can never be, or monera can never be multicellular. They are always what? They are always unicellular. Tell me, guys, is it clear or not? Is that clear? Martin, Yashir, Nabiha,
Yes, sir.
Yes, sir.
clear? Haya, is it clear now?
Yes, sir.
Great. So you'll get the link of PPT. Now we have to start with the. So I have told you already that in kingdom Monera, here, look, this is bacteria. So all the organisms are made up of prokaryotic cell. Here you will not find the nucleus, right? All the organisms are made up of prokaryotic cell, right? So what are the, which organism come under this Monera? So under this Monera, I have already told you there will be bacteria, right? So there will be eubacteria. This is eubacteria, means new bacteria. Archaebacteria means older bacteria, and there are again the next representative organism, lubrine algae and mycoplasma. I will recap, give extra class for the new student, right? Even the older, if you want to know, if you want to join, you are most welcome. And here, then I have said that I have started with the protista. So the kingdom protista is called mixed kingdom, right? The kingdom protista is made up of all unicellular eukaryotic cell, right? Then I have again divided kingdom, means the scientists have again divided the kingdom in three categories, right? So protista can be of three categories. First, photosynthetic protista. Those protista which can make their own food, means they are green. Consumer decompose means those protista which are like fungi. They decompose dead and decaying material and take their food. Third one, protozoan protista, which are like animal-like cell. They eat something. Neither they can make their own food, nor they can decompose it. Is it clear now? Is it clear to all of you?
Different types of protestant, right?
Yes, they are a type of protestants, right? So we have again divided protestant into three categories. Fine. Now we will study one by one, right? First, we are going to study photosynthetic protestant. Means those protestant which make their own food. Means those unicellular eukaryotic organisms which are capable of making their own food. So first kind of protestant, what are the first kind of protestant? So first kind of protestant, they are the photosynthetic protestant. So photosynthetic protestant, dinoflagellate, chrysophyte, and euglena. First, dinoflagellate. Okay. So these are protestants, all these three, dinoflagellate, chrysophyte, and euglena. They can make their own food, right? So collectively, all these three, they are called phytoplankton. Why? Phyto means plant, and plankton means a small plant-like organism which float on the ocean. What is the use of this phytoplankton? Basically, those those fishes which are vegetarian fishes, they generally eat on these phytoplankton. Small fishes, they eat these small plant-like organisms and take their. Then some crab-like organism, crab, crustaceans, which is small insect which are found on the oceans. They eat them as their food, right? So all these organisms means dinoflagellate, chrysophyte, and euglena mean photosynthetic protistans are called phytoplankton. Next, they are green and photosynthetic organisms means they all are green. They can make their own. Now, the first type of the photosynthetic protestant is dinoflagellate. Dinoflagellate, the example of dinoflagellate here, the name of this example is goniolex. The cell which is visible, or we can say the organism which is visible to the in your screen, this name is goniolex, right? Goniolex is a type of dinoflagellate, right? What is the peculiarity? They have two flagella. This is the one flagella, which is this flagella. What is the use of flagella? It's hair-like structure, and they use it for swimming, right? There is one horizontal flagella and one vertical flagella. Look, this is the then second flagella. So they have two flagella and both are at the 90-degree angle, right? They have some nucleus. They have nucleus, right? Means nucleus is present because they are eukaryotic cell, right? There is this girdle-like structure. This girdle-like structure. This girdle-like structure is called cingulum. They have oil droplet. Oil inclusion means oil droplet, which is as a reserve food. They have starch, which is collected in the form of food. So this is the structure of this dinoflagellus, means goniolex. What is their characteristic? What is their general characteristic? General characteristic is that they are mostly marine, means most of them are found in sea water or ocean water, right? In salt water. But some are found in fresh water. Very few of them are found in lake and river where sweet water or soft water is present. Second, there are two examples of dinoflagellates, gymnodium and goniolex. The goniolex and gymnodium, the diagram of both cells is here. Look, this is goniolex, this one is goniolex, and this one is gymnodium, right? So gymnodium, this is gymnodium, this is goniolex, right? What is the peculiarity? Gymnodium and goniolex, uh, they make the water look red because if they are present in the water, water will look like red, right? And if the water look like red, we call this phenomena red tide, right? So that this is called red tide. They give water red appearance because their color is red. Some of them means some of dinoflagellate, they shows the phenomena of bioluminescence. What is the bioluminescence? Means they emit the light. So at the night time, if they are present in the water will appear glittering. Means like you will feel that the light is coming out of the water, right? Like you must have seen the firefly. It appears, it's blink like something, like it emit the light. So can anybody tell me why the firefly emit the light? How it emit the light? Does it have some electrical charges or electrical bulb like thing? Firefly you must have seen. I think all of you must have seen.
Yeah.
Can you tell me, Martin, why it emit the light?
I don't know, sir.
Don't know? Yasir, can you tell me why the firefly emit the light? Nabiya? Anyone of you have idea?
Because of the bioluminescent phenomenon.
Yes, how does it appear bioluminescence phenomena? That's true. Tell me, Shidika. How did they produce the bioluminescence? You know,
I don't know,
when
sir.
okay, so I'm going to give you a very important information. You know, these firefly, they excrete out their excretory product, means their waste product. When they excrete out this waste product out of their body, a reaction takes place between the air and the excretory product. And because of that excretory product, during the excretion, they produce the light. Right? They produce the light. Okay. So cells are basically covered with coat, right? Means shells are covered with, you can see there are plate like structure. So these cells are covered with a covering, a coat, right? This covering is called lorica or theca. You have to remember this thing, right? This covering, you can see the covering is outside. So this covering is called lorica or theca, right? Covering, you can see this covering, this outside shield, right? Outside cover, right? Which look like a fish, like a scale, right? So the cover which look like a scale, this cover is called theca or lorica, right? And this theca or lorica, it protective shield or protective covering, it is made up of what? It is made up of cellulose, right? Theca or lorica is made up of cellulose. Fine. They have it is made up of cellulose. They are also called golden brown protistans because their color is golden brown, right? They have two flagella, one horizontal, one vertical. I have already told you, you can see, look, this is the transverse flagella. Try to understand. This is the transverse flagella, right? And this is the longitudinal flagella. So I have already told you that they have two flagella at the angle of 90 degrees, right? They have two flagella. They contain chlorophyll A and chlorophyll C. Chlorophyll B is not found, which is a part of these higher plant. Angiosperm. Starch and oil food as a reserve food in their body is starch and oil. Asexual reproduction takes place through cell division, right? They divide normally asexually by cell division. Sexual reproduction is found in some. Tell me, is it clear or not? Dinoflagellate clear or not? Yasir, Nabiya?
Found in fresh water.
Sorry,
Found in fresh water.
Martin, I'm not getting you. What do you say?
Where is dinoflagellate found?
Dinoflagellates, they are the part of this one, photosynthetic protista. Yes.
Yes, sir.
Okay. So then we will go start with the chrysophytes. What about the new student? Is it clear all of you or not? Tell me. Yashir, Nabiya, you can text me. Great, great.
Yes,
Okay,
sir.
Yashir. Fine, fine. Any one of you, if you feel problem. Now the second number is chrysophyte. So I have told you that there are three dinoflagellate. There are three photosynthetic protista. Dinoflagellate. Now we are going to study with the chrysophytes. What are the chrysophytes? So this is a diagram of chrysophyte, right? The chrysophyte, they are like your tiffin box. They are very small, but their structure is like your lunch box or tiffin box, which you take in your school time, right? When you were a small kid, you used to take a tiffin box or lunch box, and that lunch box have two covering. One part which fit on another part, right? So that kind of the same kind. So there are two parts. This upper part is called epitheca and lower part is called hypotheca, right? And outside there are some design or groups. These are called girdle or bands, right? There is a central structure. This central structure is called raphe, right? Raphe. This covering, outer covering is called frustule, right? Frustule, right? Frustule, what is this outer covering? This complete outer covering is called frustule. This outer covering is made up of silica. right? It is made up of silica. That's why the dye, they do not degrade, right? They do not degrade. Okay, so they are just these kind of the simple kind of the organism. Normally, what is their name? Normally, they are known as the diatom, right? That is the important thing. So, what is their common name? So, basically, they are chrysophytes, and normally, the example of chrysophytes is diatom, diatom, right? Or desmids. Desmids, right? Diatoms or desmids. Now, what are the diatoms or desmids? Look, uh, they all are aquatic.
So where do we start writing?
Sorry,
Where do we start writing?
I'm not getting you.
Where do we start writing from?
Okay, should I write a bigger one?
No, like do we write now? Like do we write in the notebook?
You can, your voice is not clear actually. You can
Can
text
we,
me.
should we write a notebook?
No. You need not to write in the notebook as usual. Like in the previous class, I am writing over here, right? Okay,
Yeah.
you'll get the just link. Fine, and whatever I will
Yes,
dictate, that you have to write it down. Fine.
okay.
Okay. Okay. So look, they all are aquatic, and moist, terrestrial habitat. Means where they will be found. They will be found either in the water or in the moist habitat, right? Okay. And the second thing is that how they move. So they move like this one, like, so they glide because they do not have flagella for locomotion. So how they will travel from one point to another point in water, right? They will start gliding movement. What is the gliding movement? Like they are jumping like this one. They contract, then they will inflate, contract, inflate, or contract, inflate. That's how they will jump, right? So they jump like this one, right? So this shows gliding movement. This shows gliding movement, right? That's how they show, right? This is the mode of locomotion, right? Locomotion means how to travel from one point to another point, right? What happened? Uh, their frustule, their frustule do not decay easily. Decay easily. In hundredth of year, they do not decay because the silica do not decay, right? So what happened? They keep on dying and they keep on depositing on the ocean floor, right? And they make a soil-like structure, and that is called siliceous earth, right? That is very important point, right? Right. So they do not decay easily. They pile up at the bottom of water. They pile up at the bottom of water reserve, and form diatomously, diatomoid form, diatomaceous earth. Right. So they pile up at the bottom and they make diatomaceous earth. That is there. What we do that we take out this diatomaceous earth. That is very important. You have to remember diatomaceous earth. Did you got my point or not? Tell me, guys. Did you got my point?
Yes, sir.
Okay.
Can you
So
repeat?
what is the use of this sand? So what happened? Look, try to. I'm trying to explain more clearly. Suppose that this is the floor of water. This is the. Just try to understand. This is the ocean floor, right? Right. And what is that? Water is there. So this is suppose that this is the layer of water. Right? Water. It is water. Right? What is happening? There are these kind of they are the diatom, right? So what will happen? They will complete their lifespan. After that, they will die and they will fall on the ocean floor, right? They will keep on depositing on hundreds of the year, right? After that depositing, they will make a thick covering, right? So this thick covering which will be made on the base of the ocean, right? This thick covering on the base of ocean, this is called diatomaceous earth. This is the diatomaceous earth. We take out this soil or we take out this material. What we make filter from here. And what kind of the filter? We make the filter which is used to the filter the or clean the whiskey, to clean the alcohol, to clean the syrups, and to clean the oil. That kind of the filter are made from here. One more important thing, you must have used the hair gels. Have you used the hair silicone gel? Have you used that? They make your hair silky. You must have used that.
Yes, sir.
Silicone hair oil. These silicone hair are made up of this diatomaceous. It is also used in the toothpaste. When you use the white color of toothpaste, this Colgate and all that, you must have feeling there are some crystal-like structure. Those crystals are basically from the diatomaceous. So that is the use of this diatomaceous. Did you got that point? Is that clear to all of you?
Yes, sir.
Okay, great. So I'll tell you what is the economic importance of those. In the meantime, next point. They do not possess flagella. They do not have flagella. So flagella is not found there, right? Flagella is not found there. And cover, they have two cover, epitheca and hypotheca, that I have told you, right? This one, hypotheca and epitheca. So that is cleared from this diagram only, right? And reproduction. Reproduction takes place through binary fission. Means one cell will divide into two, binary fission, and they will become two cells, right? Means two organisms, they will become two, right? And what else? Just a minute. Okay, I was just creating some space. Fine. So reproduction, that was the reproduction. And economic importance, I have told you, but let me write it down. What is their economic importance? So Very, very important point. Let me make a star. About half of the organic matter, about half of the organic matter, synthesized in the world is produced by them only. What does it mean? It means across the earth, whatever the food is made by the process of photosynthesis, half of the food is made by dinophytes. This one, chrysophytes, and diatoms. Is that clear? This is very important point. Half of it. You can't imagine these teeny kind of the cells. They make half of the food of the world, right? Whatever the half of the food, whatever the
Who
food made
makes
on the
half food?
earth.
Who makes half food of world?
Yes, half of the photosynthesis is done by them, and rest half by the plant, all the plants. Got it?
What's it called again?
Sorry,
What's it called again?
like organic food, that organic food, diatoms, these cells, these chrysophytes, or diatom,
Okay.
they make half of the food of the world, and rest half is made by the green plant.
Oh,
Got
yeah,
it?
okay.
Okay. Now the next thing is that that diatomite, this diatomite earth, diatomite earth is used as cleaning agent in toothpaste, comma, sugar, alcohol, oil, syrup, etc. Right? So this is used over there, right? And they are also pollution indicator. Last point. They are pollution indicator. Tell me, guys, is it clear or not?
Yes, sir.
Sir, I didn't
Yes.
understand the last point.
Sorry. Last point. They are pollution indicator. If water will become polluted, right, they will die.
Okay.
Tell me, I want the response from new one. Yashir, is it clear? Is everything clear to you? Nabiha and Haya. Okay, Yashir is clear. What's about you, Haya? Please tell me. You can text me if you are unable to unmute. Shidika, clear? Martin, clear?
Yes,
Okay.
sir.
Great. You know, this is a bit uninteresting part, but you have to do this, right? So that's why I am frequently asking you, is it clear or not, right? Because you know these are the organisms which are not generally we see in our daily life, right? So that's why it is really hard for us to internalize, right? The third one is euglenoid, right? Third one is euglenoid. So this is euglena, right? What is the specialty with euglena? Can you tell me? Any one of you? Always try to tell me the answer.
Tail, tail,
What is the peculiarity with euglena?
tail, tail,
What is pellicle?
tail, tail.
Pellicle. This one. They have pellicle. Tail.
Yeah.
Okay. Yes, Shidika, tell me. Will you try? Haya, Nabiya, Yashir, will you try? This is the single piece in this earth, right? There is a no second organism which matches with this one. There is a single piece in this earth, right? Which have the special quality. This kind of the quality is not found and in any organism across the world. There are the billions of the type of the organism in this earth, but no one is like Euglena. Have you seen any animal who can make its own food?
Plant, no, animal. No.
This is the only animal cell which can make its own. This is the animal cell, but it do not require food from outside. First, if light is present, it will make its own food. But if light is not present, then it will eat food from outside, right? So that's why this is called connecting link between plant and animal, right? So they are called connecting link between plant and animal, right? So first point. So this is the example of euglena. Euglenoid. Euglenoid is a class, and euglena is an example, right? So example is euglena. This cell is this diagram is belongs to euglena. First, let me explain you about this diagram. So in this diagram, you can see there is a nucleus. Obviously, it's eukaryotic cell, so nucleus should be over there, right? This is the nucleus, right? And this is a nucleus, and this is a nucleolus inside nucleus, right? Mitochondria. Look, there is a chloroplast. No animal cell can have chloroplast, but this is the animal cell which have chloroplast. It have flagellum, which help in swimming, right? And there is a this is stigma or eye spot. Stigma or eye spot provide the sense of the light, right? They provide the sense of the light. If the light is present, they will get the sense from the stigma or eye spot, and they will start floating on the surface to get the light for photosynthesis. If the sunlight is not there, they will get the sense from the stigma or eye spot. They will go at the bottom of the lake, right? There is a contractile vacuole. This contractile vacuole is a nature of, it is a characteristic of animal cell, right? You can say what is the characteristic of animal cell, right? Chloroplast is a characteristic of plant cell, right? One more important, their outer covering, this outer covering is called pellicle. Pellicle is characteristic of animal cell, right? Pellicle is not found in the plant cell. Now you can ask me what is the pellicle. So pellicle is covering. This is a covering made up of protein. Made up of protein, and it's stretchable. It is stretchable, right? You can stretch it, right? So this is an stretchable covering, right? This is the characteristic of animal cell. The same diagram is over here. Pyronoid, what is the pyronoid? Pyronoid are some bodies which collect the food. So these are the food storage body, right? Storage body. These are the body in which the food is stored, right? Chloroplast is already found. We have the eye spot or stigma, nucleus, contractile vacuole, photoreceptor. The same thing is given, right? So this is the only animal cell which can make its own food. That's why we call This is connecting link between plant and animal. Did you got my point? This euglena is called connecting link between plant and animals. This is the only animal cell which can make its own food, its own by photosynthesis process, right? So this is the on that's why this is called connecting link between plant and animal. This is important. So I always make this star in front of those which is important, right? This is it have pellicle, body is covered by pellicle, body is covered with pellicle. I have already told you what is pellicle. Pellicle in place of cell wall. Okay. Now where they are found, right? What is their so they are found in, they are not marine, they are found in fresh water. When I say fresh water means which is not salty water, right? It is soft water like the river, lake, pond. That all water is called fresh water. Fresh water doesn't mean filtered water and all that, right? Fresh water means which is not salty water, right? It may be dirty or clean, whatever, but they are found in the fresh water, right? Okay. What is the next thing? They have flagella. They have flagella for locomotion. Guys, if you have any confusion at any point, you can ask me, right? If you had never hesitate asking any simple question or simple concept or simple term, right? Always clear here right now. So they have two nucleus. No, they have single nucleus. Some of them have two nucleus. So one more important, right? What kind of the nutrition they have? They have mixotrophic nutrition. Mixotrophic. What is mixotroph? Or what is mixotrophic nutrition? Try to understand. In mixotrophic nutrition, what happened? They make their own food in presence of light, right? If light is present, so what will do? They will do the photosynthesis. They will do the photosynthesis and make their own food. So what we will call them? We will call them autotroph. So autotroph are those which make their own food, right? If light absent, what will happen? They will eat some organic particle, right? They eat. If light is not there, what they will do? They will eat organic particle. Because if light is not there, they will not be able to make the food. If they will eat the organic particle, and they are called heterotroph, right? So then they have two kind of the nutrition. So that kind of the nutrition. So they have two kinds: autotroph plus heterotroph. So if both kind of nutrition is present in any organism, what we call them? We call them mixotroph. They have mixed kind of nutrition. Mixotroph. Is that clear or not? Tell me, guys.
This is also
Clear,
known
Martin?
as for autotroph and heterotroph, right?
Exactly. They have both kind of nutrition. So what kind of term will we use? We will call them heterotroph. Euglena is the only organism which is heterotroph or mixotroph. No other organism is mixotroph. Clear?
Yes, sir.
Okay. Okay, there is a question. No, Shidika, it have nucleus because this is eukaryotic cell, right, Shidika?
Yes, sir.
Because this is eukaryotic cell, we will call it nucleus, right? If it would have been prokaryotic, we would have called it nucleoid, right? So there is a nucleus, and inside nucleus there is a nucleolus, right?
Yes, sir.
This is small one is nucleolus, and this whole structure is nucleus. This one is nucleus. So inside nucleus, this is nucleus. So inside the nucleus there is a nucleolus, which is called factory of ribosome. Formation of ribosome takes place here in nucleolus. We'll study in cell biology part, right? I'll tell you. Fine. So this was the peculiarity of this one, and they have flagella and reserve food. Reproduction takes place through binary physics. They will divide into two and make two new organs, right? So reproduction takes place through binary physics. And pellicle I have told you. And yes, one more important thing, you can see this contractile vacuum. So what is the contractile vacuum? Contractile vacuoles are organelle which help in the excretion, right? So they have contractile vacuole which help in excretion, right? Which help in excretion. So this was all about the euglenoid. Tell me, guys, if you have any confusion, any in any point, please ask me. Then we'll go to the next one.
Sir, do all animals have the euglenoid?
All, all
one?
animals.
Do all of them have the cell?
This one,
Euglena?
I'm not getting your point. Yes, tell me again. Does all animal have?
I sent it in chat, sir.
Okay. All animals have. No. Look. All unicellular animals are not euglenoid. Look. Try to understand me. We have divided photosynthetic protista in three categories. Dinoflagellate, they are purely plant. They are purely plants. Got it?
Yes.
Chrysophytes. In chrysophyte, they are also very like plant cell. Got it? Euglenoid.
Yes.
They are animal cell, but have capability of make. They have some characteristic of plant as well. Got it?
Okay, sir. Yes, I
All
got it.
euglenoid made up of animal cell, but those animal cell have some characteristic of plant. What is that characteristic? Chloroplast and making their own food.
Okay, sir.
Means they are not purely animals. Purely animals. Got it?
Yes, sir.
Nabiha, clear? Yasir?
Yes, sir.
Aya? All clear?
Yes, sir.
Okay, great. So look, let me again recap. So what I said, that photosynthetic protistans, they are like protistans have been divided in three categories. Photosynthetic protestant. So photosynthetic protestant, again we divided them into three categories, right? What are those categories? First, second, and third. So what are those categories? Look, we have divided them in three categories: dinoflagellate, chrysophyte, and euglenoid, right? So dinoflagellate, chrysophyte, and euglenoid. Again, the second one are called composer, consumer, and decomposer. They are basically, they resemble with the fungi. We have taken out them from fungi, right? So there are only one type of the dinoflagellate decomposer. They are called slime mold. Slime mold. What are the slime mold? Try to understand, right? So in photosynthetic protection, there are three categories. In consumer decomposer, they are just single category, and in protozoans, there will be again four categories, right? So photosynthetic protection. Now come to the composer decompose. So they are very simple kind of the cell, right? They are called slime mold. They are very small organism, and there are two kind of the protoplasm. Outer peripheral protoplasm is called ectoplasm. Inner protoplasm is endoplasm, and there are multiple nucleus, right? What we call when any single cell have multiple nucleus, right? So this condition is called xenocytic condition. Xenocytic condition. So what is the xenocytic condition? When any single organism have multiple nucleus, we call this xenocytic condition, right? They can change their shape. They are found in the grassland, right? And normally they cause many diseases, right? Look, this is their life cycle. What happened? This is they look like this one. This slime mold look like this one, feeding plasmodium. So they keep on feeding organic matter. They keep on feeding some protozoa and all that. They eat the protozoa as well, right? Then what happened? They get mature. This is called mature plasmodium, right? This is mature. They get mature, right? And after that, they form this kind of this structure. This is called sporangium, right? Then this sporangium become larger. This is stalk, and here mitosis takes place. They make number of the spores, right? What happened? These spores germinate. After that, after germination, they rupture. There comes some amoeba like cells. These amoeboid cells fuse, and they make, so fusion of cell is called syngamy. Syngamy means fusion of cell. Fusion of cell. So fusion of cell takes place. Again, they make a zygote diploid cell, and then again mitosis takes place. They make this structure. That is their life cycle. So they are very simple kind of the organism. They are very small organism, and they are not that much tough. They are found in the grasslands and all that, and even they melt with the heat of the sun as well, right? So is that clear? Tell me.
Yes, sir.
So let me write it down. Their characteristic. What are their characteristics? they are also called fungal animal, right? Why? Because they are decomposer, but they eat some small protozoa and all that. So eating organism is characteristic of animal, right? And they are decomposer, that is the characteristic of fungi. They have the cell wall, that is the characteristic of fungi. Sorry, they do not have cell wall. They are decomposer, so that is the characteristic of fungi, right? So look, they are called fungus animal. Fungus animal, because they possess the characteristic of both. So slime mold, slime mold possess characteristic of both animal and fungi, animal and fungi. Therefore, they are called fungal animal. Fungal animal. That's why we call them fungal animal, because they have the characteristic of both, right? And they are also called, they belong to kingdom Protista. They are also called Protestant fungi. They are also called Protestant fungi because they are like fungus and they belong to Protista. Right? They do not have chlorophyll. They means they are not green. They do not have chlorophyll. Right? Chlorophyll is not formed. They do not have cell wall. Right? The body is surrounded by plasma membrane. They are found. They are found decaying vegetable. Decaying vegetable. And lawns of grass. Lawn of grasses. Right. Now, what kind of the nutrition they have? Their nutrition is of two types, right? Sometimes they behave like animal. That's why they are called fungal animal, right? So they are phagotrophic. Do you know what is the phagotrophic nutrition? They have phagotrophic nutrition. What does mean by phagotrophic nutrition? Suppose that this is the slime mold. Try to understand me, and in case if you didn't get, tell me that you didn't get. Suppose that this is an cell, right? And there is this is the food particle. Right? What will it do? This food particle, it will engulf this food particle, right? It will change its shape and it will engulf this food particle. This kind of nutrition is called phagotrophic. Phagotrophy means cell eating. Cell eating. And this
Sir,
is the
could you scroll up a
characteristic.
little bit?
Sorry.
Could you scroll up a little bit?
Animal. This is the animal characteristic, right? Suppose that this is a food. This is a food particle, right? And this is slime
Yeah.
mold.
No, sir. Could you scroll up?
This is called
Okay.
phagotrophic nutrition. We call it phagotrophy. Means cell eating. Phagotrophic means cell eating.
Yes, sir.
Got it?
Could I see the earlier points that you wrote?
This point, this one, is it? Does it? Isn't visible to you? This one.
Yeah, sir. There.
Okay.
Thank you.
You will get the link of this one, right? You can read it, right? They will give you link.
Yes.
My team will
Yes,
give you
sir.
link. You can read it, right? So this is try to understand, right? Phagotrophic means phago means eat. Phago means eating something, right? So even we are doing the phagotrophy. Phagotrophy means when something is eating. Phagotrophic means basically cell eating. So like amoeba eat like this way, right? So this is food and this is slime mold. So when slime mold, this is called cell eating, right? And the second kind of the nutrition is saprophytic nutrition. Saprophytic, you know what kind of the nutrition. When any organism, so second is saprophytic. Saprophytic nutrition is characteristic of what? Fungi, right? In saprophytic nutrition, they are found on the dead and decaying vegetable. They release their enzyme and outside, right? And absorb nutrition from dead and decaying vegetable. Decaying vegetable. So look, saprophytic nutrition is a quality of a characteristic of fungi, and phagotrophic nutrition is a characteristic of animal, right? So they have both kind of the nutrition, phagotrophic as well as saprophytic. Is that clear or not? Tell me. Tell me if you have any doubt.
I got it, sir.
Martin? Yes, Martin? Clear? Nabiya, clear? Shidika?
Yes, sir. Clear.
Okay. Martin, are you saying something? Did you say
Yes,
something?
sir.
Okay. So,
What do you think, sir?
yes, Martin,
What do you say, sir?
did you said something? Are you asking something? Any doubt?
No.
Oh, fine. So, slime molds resemble with both protozoa and fungi, right? So that's why they are called animals or fungal animal, right? And reproduction is both kind, right? So sexual as well as you can see here, right? Both. Look, this reproduction, spore formation, this is asexual reproduction. Asexual reproduction. And here, what is that? Two cells are fusing with, this is the sexual reproduction. So both kind of the reproduction is present. Both sexual as well as asexual reproduction. Asexual reproduction is found in slime mold. Right. This is all about this slime mold. Tell me if you have any doubt. Ask me right now. Then we will move to the protozoa. Guys, clear?
Yes, sir, it's
Yeah,
clear.
yeah,
Great, great.
yes.
Will be okay, Shadika.
No, sir, I'm done with it. I'm done.
Okay, fine. After the class, please revise it, right? And because if you will not revise this part, I'm more than sure you will miss it, right? Because if you think that yes, oh, it's okay, I'll revise it after 2 days or 3 days, you'll miss it. It is a bit complex part, and believe on me, if you get this classification system, plant classification is easy, animal classification is easy. Uh, Protista, Monera, and Panjai. These three classification system, they are a bit complex. If you'll get this classification system, then everything in this class, your 11th class is easy. And 12th class syllabus is easier than 11th class syllabus. In 12th class, you have 16 chapter in biology, but in 11th class, you have 21 chapter. So there is a huge difference between the number of chapter as well and the syllabus as well, right? So 11th class syllabus is a bit complex, but 12th class syllabus is a bit easy. So here you have to revise everything again and again. In case if you are facing that I didn't got this chapter or I didn't got this topic, I am there to make you understand again with another logic, with another way, like, to make you understand. But believe on me, if you are ready to get get the any topic or to get the any chapter, so that is my guarantee. I'll make you understand. But provided you must be ready to like with like by your heart, means you must be ready with by your heart to learn, right? Then I'll make you learn. So this is my recommendation. After the class, when you get the link, revise it again, right? So look, what I told you, that the types of protestant, there are three kind of photosynthetic protestant. I told you dinoflagellate, chrysophytes, and euglenoid. Then it comes to the this one, uh, consumer decomposer. There is a no type, there is only one type, that is a slime. Now when we are talking about the protistants, right, they are basically the animal cells, right? So there are basically three kind of the protistants, sorry, four kind of the protozoa, right? Protozoa and protestant. So protozoa are basically the plant cells, animal cells, purely animal cells, right? So first, flagellated protozoa, those protozoa, protozoa, those protozoa which have flagella, right? The second one, amoeboid protozoa, amoeboid protozoa. Third one, sporozoans. So there are three kind of flagellated, amoeboid, and sporozoan, right? So now we are going to study the protozoa. Right? So protozoans, as I have already told you, that there are three kind of the protozoans, right? So normally first come to the protozoa. What is the protozoa? So this is the cell of, this is the amoeba. Amoeba is represented of protozoa, right? So protozoa, they are basically animal cells. They are basically unicellular animals, right? So normally they are unicellular. They have all characteristic of animal, right? And they were first studied by a scientist, and the name of they were first studied by Leeuwenhoek. They were first studied by Leeuwenhoek, right? And flagellated protozoa or amoeboid protozoa. That's true. Third one is protozoa. Yes, there are four categories. That's what I was thinking. There are four categories of protozoa. Ciliated protozoa. So the fourth one is ciliated protozoa. Ciliated. So further, protozoans can be divided into, there are, four type of protozoa. Four type of protozoans. The first one is, that is amoeboid protozoa. Or flagellated protozoa. Those have the flagella for locomotion. Second one, which look like this one, amoeba. Second one, they are amoeboid protozoa. Right. The third one, that is the sporozoid protozoa, right? Like malaria parasite. They are called sporozoans. The fourth one, they are called ciliated protozoa, like paramecium. Ciliated protozoa. So let's start with the amoeboid protozoa. Right? When I talk about the amoeboid protozoa, it means we are talking about the amoeba, right? So amoeba, you can see the first one. So let's start with the first one. Rather than amoeboid protozoa, I should start with the flagellated protozoa. Flagellated protozoans are those which have the flagella for their locomotion. So this is also the representative organism of this one, protozoa, right? Amoeba. So let's start with the flagellated protozoa. Flagellated protozoa. The example of flagellated protozoan is Trypanosoma gamians, right? What is the Trypanosoma gamians? Trypanosoma gamians is a kind of the organism which have uh, responsible for a disease called sleeping sickness, right? So the sleeping sickness kind of the disease is caused by this organism. Let me show you the diagram of this one. Protostome, flagellated protozoan. Where is Trypanosoma? Yes. Yes, this is the diagram of Trypanosoma gamete. This organism, it causes a kind of disease, and that is called sleeping sickness disease, right? What is this? This is the most common disease, in one of the most common dangerous disease, which is found in the African continent, right? This one, which is called sleeping sickness. Like a person which is infected by this organism goes into the coma, right? So today, like we have limited time, so we'll start with this protozoans in the next class. So basically, look, try to understand. They are the animal cell. They engulf the food, right? So their nutrition is basically the phagocytic kind of the nutrition, right? They have the phagocytic nutrition. As you can see, they look like amoeba is the ideal protozoa. So protozoa is basically represented by the amoeba. So they have this foot-like structure. They are called pseudopodia. Pseudo means false and podia means foot. So they have false foot. Their nucleus is present. Obviously, they are eukaryotic cells, so nucleus should be. They have the food vacuole. After engulf the food, they keep the food in a vacuole-like structure. This is called food vacuole. There are contractile vacuole. I have already told you that contractile vacuole is meant for excretion. And you know, the excretion, this contractile vacuole is also characteristic of animal cell, right? So cytoplasm is divided into two, ectoplasm and endoplasm. The density of endoplasm is more and ectoplasm is less, right? They have some crystal like structures as well, right? So okay, fine, Yasir, you can go, even I'm just going